Hello, everyone. It's time to get ready for another lab. So this week we're beginning a two-lab sequence working on the iodination of vanillin. So this is actually going to take 3 whole weeks to do. And one of the reasons why is because this is part of what's called a two-step linear synthesis. So linear synthesis means that I'm going to go ahead and take the product from one reaction and use this for the reactant of another reaction. This is very common in typical organic chemistry, as the species that that you need as a reagent can't actually be bought over the counter. So you have to end up making it first. So in this linear synthesis, we're going to perform two parts. The first one is going to be experiment 7, where we're going to perform the iodination of vanillin. Literally, I'm going to take vanillin, same molecule we used last week for glow sticks, and I'm going to add a single iodine to the ring. Much trickier than it might seem at first glance. Then in the second step, we're going to perform what's called a coupling reaction. So a coupling reaction is essentially a very specific combination of taking two different reagents, in this case, the iodine that we just put on our vanillin, and doing a Suzuki coupling, going ahead and attaching it to this boron position. Again, coupling together our two different species. So let's go ahead and examine the part that we're going to be doing in this experiment, specifically making that iodovanillin. So to perform this experiment, we're going to be mixing together our vanillin along with potassium iodide and oxone as our oxidizing agent in water, and we're going to pretty much reflux it for an hour. Pretty much we're going to keep it at a nice simmer the entire time. So when we do this process, our product should be our iodovanillin. So we're going to take this solid product and we're going to collect it by vacuum filtration, and then we're going to store it for a week. Because if you, as you may have learned from previous experiments, the product you get straight from a reaction is always a little bit impure. So we're going to go ahead and purify this with our classic method of recrystallization. So in week 2, while you're performing experiment 7, there's a long, again, another long, quiet reaction time that we get to sit around with. And during that time, you can go ahead and recrystallize your iodovanillin. And then we're going to use this recrystallized iodovanillin, and we're going to analyze it via two major methods. First of all, our old friend melting point, and the beginning of a new type class of method, spectroscopy. Specifically, we're going to use IR or infrared spectroscopy. All right, let's dive into the details of this lab. So first thing we're going to be doing is that oxone reagent, probably not one you've seen before. This is essentially a persulfate, where we take sulfate and it has essentially a peroxo bond attached to it. Turns out this is a really good oxidizing agent. And what it can do is activate that potassium iodide, as it can go ahead and react with the iodine. The iodine here is going to act as a nucleophile, again, attacking that electron poor hydroxyl group. This will essentially make an activated iodine hydroxyl species. At this point, the electronegativity actually favors the electrons being on the oxygen. So in practice, we can go ahead and treat this as a cationic iodine or an electronegative iodine. In truth, that iodine is going to be on a whole bunch of different compounds. It's going to get passed around. Again, we have our initial hydroxyl iodine species, but we also have other classics like molecular iodine. In each of these cases, that iodine is acting in an electronegative, in an electron poor fashion. This means that the iodine can then act as an electrophile in a sequential electrophilic addition and an electrophilic elimination reaction. These are more commonly known as electrophilic aromatic substitutions. So in this case, it's going to be my aromatic ring is going to act as the nucleophile, reach out, grab the iodine, at which point this puts a positively charged cation on the aromatic ring. This is semi-stable due to resonance, but will quickly essentially be inactivated. And we can do this by a electrophilic elimination. where a base in solution, and there are plenty in there, including sulfates, hydroxides, and iodines, can go ahead and remove this proton, again pushing those electrons back on to reform that double bond. So the only net effect we see is switching out the hydrogen atom here for an iodine atom. And this is going to be our iodination. This reaction mechanism is also known as electrophilic aromatic substitution, and it's going to be a good classic reaction you'll cover in organic too. All right, so to actually perform this reaction, we're going to want to reflux our system. And again, as you know, reflux is a fancy word for saying simmer. So we're going to be doing this because this reaction would probably take a couple days if I was going to run it at room temperature. So in order to speed up the reaction, add heat makes it easier to overcome that energy barrier. So in order to do this, we're going to assume a reflux apparatus. So we're going to start by putting our potassium iodine and our vanillin into a round bottom flask, and we're going to have them dissolved in water. We're then going to go ahead and put this over an aluminum block, which is going to be on on a hot plate, which we can then again raise and lower to apply correct heat, as well as it will contain a stir bar, which again, the hot plate can go ahead and keep the reaction stirred while heating the entire time. To this, we're going to go ahead and add what's called a Claisen adapter, which will include two major components. First, a condenser. This will essentially make sure as the system simmers that any vapor we emit doesn't really make it past the condenser. This way, as any of the gases, including water vapor, or again, that iodine intermediate essentially come off, they'll hit the condenser, condense into solids or deposit into solids in the case of iodine, or condense into water and come right back down. This will essentially prevent all of our iodine, which represents a reactive intermediate, from escaping the solution. However, we are also going to have a second component, and this is going to be an addition funnel. So the additional funnel is going to be used to slowly add oxone to our solution. And so we're going to put a dissolved mixture of oxone in this apparatus, and then slowly add by use of by the stopcock over a course of 5 minutes to slowly mix our reaction mixture. And the nice part is by having this addition funnel, this also makes sure that I don't have to leave an opening in my apparatus as I add this component, nor have to risk it ending up all over my condenser. It adds it a lot more smoothly. And this also can be used in other reactions to essentially add other reactive components over the course of the reaction, as we can slowly switch out what's in the addition funnel. All right, so once the reaction has been refluxing for an hour, we're going to cool the reaction mixture off by essentially turning off the heat, and we'll drop that heating mantle again by using the lab jack. But we're going to try and keep it stirring the entire time. Then what we're going to do is you're going to use a little DI water to rinse down the sides of the condenser, again making sure we return any of that iodine to our mixture and using the ambient heat and the stir to help complete the reaction. Once you've done that, we're going to remove the condenser and then and the additional funnel, and again rinse down our adapter, moving all of the iodine back into our reaction mixture. Once this is done, we're going to remove that Claisen adapter and test the mixture for how much of that I2 is, to see if any of the iodine is still in excess. If it is, which it probably will be on the first time, we're going to neutralize it with some sodium bisulfate. So sodium bisulfate is going to essentially act as the opposite side of the oxone. This time that sulfate-derived group is going to act as a reducing agent. So essentially returning it into sulfate and eliminating any extra iodine. Now, after you added the bisulfate, test again. If it still contains iodine, add more bisulfate and rinse and repeat till the system is completely neutralized, as we want to remove that iodine from our reaction mixture for the future steps. Once that's done, it's time to essentially filter our product. So we're going to go ahead and dump our product onto a vacuum filter, and we're going to use DI water as our rinsing solvent, as the product should be minimally soluble in water. Then after we're done letting the vacuum filter run, we're going to let the sample dry on the filter paper for about another 5 minutes. And then we're going to go ahead and store that sample in an Erlenmeyer flask. One of the reasons why we're going to do this is just for context. In week 2, we're then going to redissolve our sample using the bare minimum of an isopropanol ethyl acetate. So again, a little bit more, a little bit lower in the polarity. This should help completely dissolve our sample. Then we're going to use hot DI water as our crystallizing solvent. Again, the reason we're going to do this is so we have to use the minimum amount of DI water to essentially get our, to mix into this less polar solution and to essentially guarantee that the maximum amount of our iodovanillin falls out of solution. Then we'll go ahead and vacuum filter the product again. So again, all of this is going to happen again in week 2 in the middle of our experiment number 6. So from this, we're going to use our dried mass of the product. You're going to let the system dry on the filter paper again, to obtain a dry mass and put in a labeled bottle for further use. All right, so once we're done with our purified product, we're going to be doing several major tests. So first thing we're going to go ahead and do is collect another melting point. So again, classic melting point of iodovanillin. Smaller the melting point range, the more pure. Wider the range, less pure. We're also going to take an IR spectra. So for IR spectroscopy, this essentially provides crucial information about what functional groups are present in your sample. This can be used to identify that we have the correct product. It should have peaks corresponding to an aldehyde, an alcohol or phenol, and a ether, and also for an iodine group. From here, we can go ahead, and if any extra peaks show up, this should essentially correspond to any impurities. You're also going to be provided a generic iodovanillin sample, so you do not need to take an NMR of yours, that we're going to evaluate using NMR spectroscopy. So again, NMR spectroscopy is really useful about providing information about what H atoms are present in solution, because based on the environment and functional groups of each of these hydrogen atoms, we will end up having a peak in a different part of the NMR spectra. And the size of these peaks will correspond to exactly how many of these H atoms are on any specific group. So we can again use this HNMR to go ahead and provide 2 major pieces of information. A, confirm our product identity, and if any additional peaks show up, this should essentially confirm if any impurities are present. However, for a full description of these 2 methods, there's going to be 2 sets of videos available on Canvas. One talking about IR spectroscopy, one set about IR spectroscopy, and one about NMR. So to prepare for lab, you're going to want to go ahead and read the Morgue chapters. So again, 21 and 22, this covers IR and NMR. You're also going to want to read the lab manual for experiment 7. You're already watching this video, so congratulations, you're almost done. And complete the pre-lab quiz. Since this lab covers mostly pre-existing material, the pre-lab quizzes in this case is going to be more or less a core survey, letting me know how I'm doing. Also, don't forget to write your pre-lab notebook and answer all of the questions to consider. Watch out, one of those is on IR spectroscopy. For actual lab, fairly straightforward. We're going to meet in the chem lounge, be present for safety presentations, set up your round bottom flask, then assemble the apparatus around the flask. Now, if you're waiting for use of a balance, to put everything into the round bottom, you can go ahead and already set up the heating mantle and lab jacket first. Once you have the entire apparatus assembled, go ahead and turn on that condenser, get it going ahead of time. Then we're going to go ahead and slowly add that oxone over the course of 5 minutes. So essentially adding it dropwise. Then we're going to reflex the whole solution for an entire hour. Once that is done, you're going to turn off the heat, but again, keep the mixture stirring. So you can't drop that heating mantle so far lest you lose that stir. Then you're going to again rinse down that iodine into the round bottom flask, test for excess I2, add bisulfate to neutralize if present. Uh, After you've added the bisulfate, test again, add more bisulfate until it is completely neutralized. Then we're going to vacuum filter that product again with DI water and allow to dry. Then you're going to more or less clean up as everything's going on around while everything's drying. Then you're going to go ahead and place that iota vanillin in a labeled Erlenmeyer, store until next week, and finish cleaning up. All right. So some reminders as you're performing all of this. So first of all, make sure to include a stir bar in your round bottom. Hard to add later. Once the apparatus is assembled, keep that condenser running the entire time. Again, while you're running this, while you're refluxing the system, make note of when I2 crystals appear. This will help illustrate that your system is readily reacting, so it is fully reacting, and note when they appear and where on the apparatus they form. Sorry, everything managed to jam. All right. And again, remember the timing for the reflux starts exactly when the solution begins to simmer, and you're going to run the hour from that point. And again, try and keep that heat adjusted so that solution remains at a low simmer for that entire time. Don't let it go to full boil, but don't let it stop simmering. And again, for that the starch test, this is going to work kind of like using pH paper, except for this time, a blue-black color indicates the presence of iodine. If the system tests positive, keep adding more bisulfate. And again, don't forget at the end to leave that product on the filter for that extra 5 to 10 minutes to finish drying as much as possible before you transfer it. Now, once you're done, go ahead and make sure to do those post-lab notebook scans as soon as possible. Again, you do not need to watch the videos before lab. Helps if you do, but the IR and NMR isn't going to be due until next week. While in lab, I am going to hand out a Experiment 7 report sheet, but do be aware it's not going to be due for 2 weeks, as you're still going to need to know your dry weight after the recrystallization to figure out things like percent yield. But you can work on it ahead of time to help be prepared. So that will be this week in lab. Look forward to seeing you there, and take care.